Today I will be talking about bee microbiomes, particularly how a space and a species is called the Stingless bee microbiomes. So it has been shown that microbiome plays an important role in eusocial bee health and fitness, but such benefits rely on a specific and balanced microbial community, which are ultimately shaped by multiple mechanisms. These may include host to microbe, microbe to host, and microbe to microbe interactions. And you can see here I add some examples, but for the sake of time and having more time to explain about our model system, I will not enter in detail right now. But what is important is that just unraveling the drivers of microbiome assembly is that we will be able to guide conservation strategies and also understand how the microbes and hosts are evolving. On the side of bee microbiomes, however, most research have been conducted on honeybees and bumblebees, while stingless bees that are the richest and most diverse group of eusocial bees are mostly understudied, despite being also highly eusocial, forming large perennial colonies, being used for pollination and also for the hive products in the tropical regions. The few studies that include stingless bees, however, have been showing that they share an ancestral microbiome with other eusocial bees, but we still know very little about the hive level, the superorganismal level microbiome, which actually has truly unique traits. So in this slide, you can see here in the center a picture of a stingless beehive, particularly Scaptotrigona, which is our model system. And on the side, I left a picture of a honeybee hive, since you may be more familiar and you may be able to compare and understand more about our model system. First thing you will see is that the stingless bees, they establish the colonies in this wooden box without frames, as observed for honeybees. But still, they are very compartmentalized. While in a honeybee hive, you can see even in a single frame, worker bees, larvae in development, and even food stores. In stingless bees, they have separate pods for honey and pollen that are apart from the brood, as well an additional worker caste. So social bees have the workers that do many tasks within the hive. For example, nurses take care of the hive and the brood. The foragers go out to collect food and bring back to the hive, while soldiers, in the case of stingless bees, they just stay in the entrance of the nest, taking care and like avoiding intruders, and they don't perform any activity within the hive. All of these that may affect or influence how microbes circulate within the hive. Another aspect is that stingless bees do mass provisioning. So in honeybees, you will see that workers offer larval diet every day, a little bit like progressive provisioning, while in stingless bees, they do mass provisioning, basically providing the entire amount of diet for larvae development. This allows the queen to lay the eggs and keep the cell immediately after the egg is laid, which also may influence in limiting microbial access and also support the local community in this environment. And more recently, which is also really interesting and unique among the stingless bees, is that they found that a yeast grows over this larval diet. This yeast is known as Zygosaccharomyces and is eaten by the larvae to complete the development, bringing a new layer of microbiome complexity that hasn't been seen in other eusocial bees, for example, in honeybees, where fungi are considered mostly transient. So what drives the assembly of a stingless beehive microbiome? Our first step was to conduct 16S sequencing to characterize the bacterial community in two species of Scaptotrigona, these stingless bees. We sample from different hive environments, including the food stores, the pollen and honey, the brood, which includes the diet, the larvae, and also the adult bees, nurses, foragers, and soldiers. And in our first result, just checking the richness of amplicon sequencing variants, the ASVs, we already saw a quite interesting result. As you can see in this plot here, the honey and the soldiers have the higher number of ASVs, meaning they have the richest communities. Most of these bacteria that don't even establish in other part of the hive, especially because these parts are spatially segregated of the other components. When we look at the community level, as you can see in this NMDS plot, we see that the hive components are segregated by color, meaning they have consistent and distinct microbial communities. This is easily observed and visualized in this bar plot as well, that is also organized by hive component, and each color represents a different bacterial taxa. Of course, you probably see that lactobacilli are the most prevalent group of bacteria within the hive, but especially in the food stores, brood, and bees that interact mostly with the nest. While in the adult bees, particularly foragers and soldiers, we start to see more the well-known eusocial bee core gut microbiome, which includes bifidobacteria and bombilactobacillus, while soldiers don't even have most of the lactobacilli found within the hive. But to get a better resolution of this community, especially for all these lactobacilli that were not characterized or classified at genus or species level, we decided to also move on with metagenomics. We were able to recover genomes of 9 bacteria consistently associated with different hive components. These include the bombilactobacillus and bifidobacteria that are most prevalent in adult bee guts. And we confirm they carry genes, for example, for plants polysaccharide digestion. But interesting, we also were able to recover this unclassified lactobacilli we saw with 16S, which represent novel species of Nicoleella and Acetylactobacillus. And as you can see here, they are part of this clade of Apilactobacillus that is a known group of beehive symbionts, and they are particularly abundant in the food store and especially within the brood, where it has been shown, at least in representant bacteria from honeybees, they can control, for example, spoilage and particularly pathogenic fungi. So together, all these results already hint for the nest architecture contributing for shaping the microbiome assembly, like allowing bacteria to settle in distinct niches and protect from influx of microbes, and also allowing with that for local adaptation. But of course, considering this result, we decided to move on also looking at the fungal community that is part and probably interacts with this bacteria. And we know that is very important for stingless bees, as I showed to you early in my introduction. And we did, in this case, ITS sequencing with the same samples. And as I showed to you in bacteria, here I show again an MDS plot. And we see distinct fungal community in different niches, but different than bacteria, the clustering by the colors in this plot is not so strong. Mostly larvae and diet have like particular distinct communities. This is more clearly seen in this bar plot, where we see like several colors in the different parts of the hive, for example, pollen and adult bees, suggesting a less consistent fungal community or transient community. But in the larval diet and the larvae, we see a pretty consistent yeast community, which was expected considering that Zygosaccharomyces is present there. But it was a surprise to see that consistency, considering we know that bacteria with potential antifungal effects are also particularly abundant in this environment. With that, we decided to isolate these microbes from the brood environment and see how they may be interacting. Maybe the bacteria from stingless bees don't have that antifungal role observed for the bacteria in honeybee hives. Here, what you see is our assays testing the effect of AP lactobacillus as a representative of the most abundant bacteria in the brood. Against different fungi, we included aspergillus, that is a pathogenic fungi isolated from honeybees. We also included Zygosaccharomyces, that is the stingless bee symbiont, and also a Zygosaccharomyces, that is a non-native yeast from stingless bees that we isolated from honeybees. And what you see here, first of all, is the control of the fungi growing. And now I show to you that we see when we put the bacteria lawn and lead the fungi to grow over this bacteria lawn, Apelactobacillus strongly inhibits the pathogenic fungi. And also the Zygosaccharomyces, that is not native or a symbiont of the stingless bees, but it promotes the Zygosaccharomyces yeast. One cool thing that this is just a general behavior of this bacteria. So we decided to include other bacteria we isolated from this environment and are not so abundant, including another Lactobacillus and Acetobacteriaceae, and we don't observe the same pattern of a strong inhibition of the pathogen and promotion of the symbiont, which suggests that bacteria and fungal interactions are pretty selective in this environment and involve specific microbes. So this together brings to like a suggestion of multiple mechanisms shaping stingless bee microbiome. And this goes from host to microbe interactions. For example, we see the compartmentalized nest playing a role in the mass provisioning, microbe to host interaction with the protection of the bacteria against potentially pathogenic fungi, as well microbe interactions with growth inhibition and promotion. All like a very complex network of interactions at the hive level that only can evolve with reliable transmission, and which brings me to this important behavior from stingless bees, that is when they form a new hive, different than other social bees that basically swarm only with the bees. In the stingless bees, they also transfer nice materials with the workers that are potentially contributing for the establishment and evolution not only of a good core microbiome, but a superorganismal level microbiome. With that, I would like to thank my collaborators, both in Brazil and from the Newton Lab, all the funding, and I'm happy to take questions, I believe, in the end of the seminar, the forum. Thank you so much.